Alright, class. So let's look at this diagram. You have seen this in your lab mini lecture before, and right now I want you to focus on the twitch. So I want you to focus between these two lines. And actually in this graph, on the x-axis we have time in milliseconds. On the y-axis we have the force of contraction or tension. And then the orange arrows represent stimuli. Now focusing on the twitch, look where the stimulus is. The stimulus is right here, right? That's a stimulus. Now you have the electrical stimulus first that leads to the muscle contraction. Now do you see a latent period? A latent period between the stimulus and the actual contraction? And this latent period is defined as a time lapse between the electrical stimulus and the actual muscle contraction. Really important. So again, this is showing us that you have the electrical event or the electrical stimulus first, followed by the mechanical event of contraction. All right. We also see summation. Let's focus on summation now. And in case of summation, we do see that we have increased frequency of stimulus. Therefore, the individual twitches cannot relax and they piggyback on the first one. And this results in, so for example, the second wave is stronger than the first and the third one is stronger than the second. Typical summation. Now I want you to focus on the incomplete and complete tetanus now, and we will talk about this. Now if you look at incomplete tetanus, what's happening to the frequency of stimuli? Yes, the frequency is high. So the frequency of stimulus or the stimulation frequency has increased. Therefore, we are stimulating the fiber over and over and over with electrical shocks. And that leads to decreased relaxation time between twitches. And this is called incomplete tetanus. However, if at maximum frequency, if we consider maximum frequency of stimulation, there will be zero relaxation. So there is absolutely no relaxation between the twitches, and we see a very smooth and sustained contraction, and this is called complete tetanus. Okay. Now, this concept is very important. Okay, let's talk about this. So let's go over frequency and summation again one more time. Okay. Now in red, you see cytoplasmic calcium levels. In blue, you see the force generated by the muscle. And in black, you see action potential generated in skeletal muscles. By black, I mean the tiny black lines at the bottom. Those represent action potentials. Now remember, with every action potential, what do we see? We see that with every action potential, there is more calcium that is dumped into the cytoplasm. Do you agree? And more calcium means prolonged tropomyosin's inhibition on actin and more interaction between actin and myosin, right? That leads to more contraction. It also means that calcium ATPase will take longer to remove calcium from the sarcoplasm to resequester it back into the sarcoplasmic reticulum. If another action potential arises before calcium has been resequestered back into the sarcoplasmic reticulum, that means even more calcium is in the cytosol and more contraction results. Eventually, the contractions will lead to fused tetany. That means the whole muscle cannot contract any further. It has reached its maximum contraction. So this graph is showing us how much calcium is floating around in the sarcoplasm, leading to muscle contraction. Okay. All right. Now, students often ask me, Rupa, how are our muscles used when they're exercised? Okay, let's play. Let's imagine you stepped on the treadmill on the gym and you start to run. Now, there is a finite amount of ATP in your muscle cells. As you exercise, you quickly consume that, right? As you use the ATP already in the cell to cycle your myosin heads, there is an enzyme called creatine phosphatase that takes creatine phosphate and takes a phosphate group and transfers that to ADP to recreate ATP. However, so what do we see here? We see an enzyme called creatine phosphate. The enzyme is creatine phosphatase. And this enzyme takes creatine phosphate. It takes creatine phosphate and sorry, I'm messing this up. Just give me a minute, class. Okay. So, there is an enzyme called creatine phosphatase that takes a phosphate group from creatinine and transfers it to ADP to make ATP. That's how the creatine phosphatase enzyme works. It takes a phosphate group from creatinine and transfers it to ADP to make ATP. However, the problem is that the activity of this enzyme only lasts few seconds. And remember, you are still running. Now, what will you use to generate ADP? Next in line is a breakdown of glucose that is already present in your muscle cell. So we are looking into the second panel now. Glucose can be anaerobically metabolized, which generates very little ATP. If you are running fast and not breathing well, your glucose stores will be metabolized anaerobically, and this means small amounts of ATP are generated and lactic acid will be created. The lactic acid causes your muscles to fatigue, and you also feel discomfort or soreness as the levels build up. You will ultimately stop running. However, if you are breathing well, the oxygen levels will allow you to metabolize glucose, fatty acids, and even amino acids that are delivered to your muscle cells via the bloodstream. Aerobic respiration means you are able to fully metabolize these and generate ATP. So in summary, as you run, you use your creatine phosphate supplies first. Then your muscles will use glycogen stores. These supplies can last about 20 to 30 minutes, and then you use your fatty acid oxidation for ATP generation. In all, it takes about 30 to 40 minutes of exercise to about 40 to 80% intensity to start to burn fat. Sheer sadness. All right, so first, this is the first step. Remember, this lasts only few seconds. Then you are using the glucose source, breaking it down to generate very little ATP anaerobically. However, if you are breathing well, you perform this, you perform aerobic respiration, by which you break down glucose, fatty acids, amino acids to generate a lot of ATP. Okay, so this you produce little ATP. This obviously by aerobic respiration you generate a lot of ATP. That's a key point here. All right, now how do you get more muscle mass? Well, you go to the gym and you increase your muscle mass, and that happens due to hypertrophy. That means the number of myofibrils and myofilaments increases. So actually the girth of the existing muscle cell increases. And there is another, so that's hypertrophy. Hypertrophy means you have more myofibrils and myofilaments. Now, Rupa, what is hyperplasia then? Well, hyperplasia is when you have increase in muscle fibers. And that is possible, that is actually more common at a younger age as compared to older people, because muscle cells undergo mitosis to generate new daughter cells. And the new daughter cells are called myoblasts. The problem is, as we age, the number of myoblasts decline. And the evidence that endurance training can stimulate myoblasts to divide and generate new muscle cells is not robust. But there are some articles out there that describe this event. So, myoblasts help in hyperplasia. If you have a muscle fiber, so let's say you have a fiber, muscle fiber that divides to form myoblast. So myoblast, you have two myoblasts now. And these myoblasts will then mature to new fibers, right? So you have more fibers. If you have more fibers, that hyperplasia, remember that is very, very common when we are younger. I have a very distant cousin who was very young when he had a injury in his calf muscles. But he was 8 or 9 years old at that time. And over time, as he grew older, he did gain some muscle mass there. So he did have some hyperplasia, by which there was increase in muscle fibers as he grew up. And that could happen because he was younger. All right. And also remember hypertrophy. The muscle fibers are not increasing. We are only having more of myofibrils and myofilaments in that case. All right. We will start a new video with muscle atrophy.